Great. Thank you all for being here today for our MPG primer. How could you forget? Yes.
We are very excited to have Dr. Hoon Cho here, who's an assistant professor of biomedical information and data science at Yale University. He was previously here at Broad as a Schmidt Fellow and principal investigator. He received his PhD in electrical engineering and computer science at MIT in 2009. And before that had been at Stanford, where he got both a bachelor's and master's degree in computer science with honors. He's been named a recipient of the NIH Director's Early Independence Award. Congratulations. The Cho Lab now works to create algorithmic solutions to tackle a variety of computational challenges introduced by the presence now of these large-scale heterogeneous data sets that have important privacy concerns. And so Dr. Cho is very happy to take your questions during the talk today. And so please feel free to come up to the microphone or type in the Q&A on Zoom. And thank you again for joining us all the way from Yale.
Yeah, thank you, Sarah. Thanks everyone for making here in person despite the rain. I appreciate people on Zoom as well. As Sarah mentioned, I was a Schmidt Fellow here for 4 years before I moved to Yale last September. So I'm very familiar with this space. It's always good to be back. So yeah, in this primer, I'll be talking about genomic privacy. And given my background in computer science, I'll be mostly sharing my perspectives on algorithmic and computational side of things. So I just wanted to mention that up front. In the first part of this talk, I'll be giving you an overview of privacy risks that are known to be associated with genomic data. And then in the second part, I'll talk about some of these emerging solutions based on what are called privacy enhancing technologies for addressing these risks. And throughout the talk, I'll try to both define the key concepts in these areas and also give you some vignettes of our recent work on these areas as well. So I want to first start by talking about what we mean when we say privacy and distinguish it from another closely related term, security. So privacy in general is a right to be let alone. That's a common way to define it. And it also means that an individual has control over how information about them is collected, shared, or used in different settings. On the other hand, security is generally about a property of a system that provides protection against unauthorized attempts at accessing the data or tampering with it, and also tampering with the functionality of these systems. So these are closely related, as you can tell, but one does not necessarily imply the other, and we're generally interested in developing systems that are both secure and provide protection for the privacy of the underlying individuals. And as you know, privacy is an important topic in biomedicine and genomics in general, and this is mostly due to the fact that biomedical information, it contains something that's very personal to us about our biology and health. And the thing that makes this problem so complex is that there are all these different types of biomedical data types that reveal different things about our biology. So including things like electronic health records, medical images, personal genomes, and more recently data from wearable devices. There's also these variety of omics data types, like transcriptomics, proteomics, and also metagenomic data from microbiomes. So all these different types tell us different things about human health, but at the same time, they increase the stakes for keeping all this information safe and protected from potential misuse. And this is not just hypothetical concerns. There are many real-world examples of privacy threats. You may have seen in the news a recent privacy data breach at 23andMe last year, which affected millions of users. The attackers were able to access raw genotype data, as well as health-related information like disease predisposition, carrier status, as well as genetic relative information for these users, which is quite concerning. And it's also even more concerning that the attackers were able to create these curated lists targeting specific groups of individuals, and they were selling these data on the dark web. Here's another more recent example from just a few weeks ago. I think the situation is still unfolding. There was a large ransomware attack at Change Healthcare, which is a prescription processing arm of this insurance company called United Health. It was reported that around 94% of hospitals in the United States were affected financially due to this attack. And there's still an ongoing investigation into what kinds of data had been accessed through this attack. So there are real threats. And I want to make the point that this is not just a concern in the commercial domain, those of us who are involved in academic research and genomics should also care about privacy too. And here are some reasons. So behind every data set, there are real people who could be affected by a potential privacy breach. So we have a certain amount of responsibility to look for safer ways to conduct research. And second, these privacy concerns and related regulations, they make it difficult for researchers across different institutions to collaborate and share data. So having better ways to protect privacy can help us break down these barriers and accelerate science. And lastly, if we think about converting biomedical insights from research into public health impact, for example, for supporting health-related decision-making for private individuals, we need reliable and trustworthy systems that can process private information. So having better safeguards can help us in that regard as well. So now let's talk about what's known about genomic privacy in more detail. So there are key properties of genetic data that make it unique and different from other types of personal information like passwords or online search history and what have you. These are 6 of these properties that were summarized by this review article by Naveed et al. I'll briefly go through them. So genetic data, it contains information about our health and behavior, as I mentioned. This is static kind of information, so it's mostly determined at birth, and it's hard to change. It's also very unique to each individual. So only tens of SNPs, let's say, would be enough to single out an individual from the global human population. Mystique is referring to the fact that there's still a lot that we don't know about what can be inferred from genetic information. These data have intrinsic value because they can be used for biomedical research or clinical purposes. And lastly, kinship means that when a privacy breach happens, it reveals information not just about the affected individuals, but also about their biological relatives. So when we're talking about risks and safeguards, it's important that we consider these properties of the data. And there's been a lot of literature on different ways of breaching genomic privacy. And this is a categorization that was provided in this other review article in Nature Genetics from a few years ago. And they broadly classified the attacks into 2 categories, identification and phenotype inference. I think this is fairly self-explanatory, but just to define it for clarity, identification means that you start with a supposedly anonymous data instance, and we're uncovering the real individual behind that data instance. Phenotype inference means we start with some piece of data about an individual, and we infer additional attributes that are not included in the data set, so including their health status, disease predisposition, things like that. So in the next few slides, I'll go over some of the seminal works that explore different types of privacy attacks in this domain. One of the earliest examples was from 2008, when next generation sequencing first came about. This attack demonstrated the difficulties of masking phenotypic information in genetic data. So the first personal genome that was released was from Dr. James Watson's sample. And in this release data set, variants in the ApoE gene, which is known to be linked to Alzheimer's disease, were masked due to personal requests from Dr. Watson. But just a few months later, another group of researchers showed that these were actually not really masked. You could infer these variants using nearby variants using linkage disequilibrium patterns. In other words, we could impute these genotypes. So this led to a larger window around this gene of 2 megabase pairs to be excluded from the data set. And note that this is just for hiding 1 gene. And so it's going to be really difficult if you want to hide information about a much larger array of disease-related genes that we know that are scattered across the genome. And from the same year, there was also this seminal work by Homer et al., where they showed that even aggregate level information, like minor allele frequencies and GWAS summary statistics, can reveal private information as well. And what they were able to show is that given a target subject's genome, you could compare it against these released statistics to figure out whether the person was in the underlying cohort or not. And this is also called membership inference attack. And while this information about membership might not be too concerning by itself, sometimes these study cohorts are associated with sensitive attributes like rare medical conditions. So that could lead to a potential privacy breach, and which is a major concern here. And so the intuition here behind this attack is that if the subject has some rare variants that are not really found in the general population at high frequency, then if these variants are represented highly in the released data, then it makes it more likely that the subject was in the original data set. That's the main idea. In a more recent work, people have shown that it's also possible to demonstrate this type of leakage with genomic beacon services, where we answer questions like, is there a person with this particular mutation in the underlying data set? And you can think of this as a different type of aggregate level information that's focusing on individual mutations. And the idea here is the same. By combining information across different queries, we can try to single out an individual. Another type of attack that's been receiving a lot of attention in the media a few years ago is identification attack based on public genealogy databases like GEDmatch. So these services include genetic profiles from millions of users and their genealogical relationships. And this is a great resource for recreational purposes for finding one's roots, but they can also be used for other purposes, like police officers trying to catch criminals based on the sample that they get from the crime scenes. And the main idea here is that you start with a genetic profile from a target subject, and then we query it against these databases to find out their close or distant relatives in these databases. And we know who they are, so now we can try to triangulate and figure out who the person was. Sometimes additionally using information like their demographic information, like age or rough geographic area where the person is. And there's been several papers that explore different aspects of this threat vector. So this first paper by Genomic et al. I think Melissa came here to give a talk a few months ago last year. And so this work showed that just using short tandem repeats on chromosome Y, you can predict the surname of the target individual. Later, Ehrlich et al. Showed that the majority of individuals of European descent are susceptible to an identity inference attack like the one that I explained. And this is indeed how the Golden State Killer in San Francisco was caught, if you're familiar with that story. And more recently, Ney et al. Showed that it's actually not just the target subject whose identity could be revealed. The individuals in the database itself could also have their genomes leaked through what's being revealed by these services. And they also demonstrated that one can insert these artificial samples into these databases to misdirect police investigators who are trying to find the criminal. So going beyond genotype profiles, there's also leakage in other functional genomic data that we need to think about. So I'm referring to gene expression data, protein expression, things like that. The idea here is that even though these other types of molecular measurements tend to be a lot noisier, they contain enough identifying information so that we can link samples from the same individual across different data sources. And this is called a linkage attack. And going beyond that, we can also try to predict genotypes from these data through what are called quantitative trait loci, or QTLs. These are genetic variants that are correlated with other molecular measurements. And by predicting genotypes of the person based on their omics data, we can also create a linkage between their genetic profile data in one data set against another omics data in a different data set. And as I mentioned, these possibilities have been demonstrated for different types of data, so including transcriptomic, proteomics, and microbiome data. And next, I want to describe a little bit about our recent work from last year in genome research, where we try to study this question of this topic of transcriptomic data privacy a little better. So again, we're interested in looking at these quantitative trait loci, but for gene expression. So these are called eQTLs. And the concern is that a potential attacker can start with the gene expression level that they've obtained and try to guess the genotypes of that person. And there are many of these eQTLs in the genome. So I think it's known that the majority of human genes have some eQTL associated with them. So you can imagine extracting a substantial portion of one's DNA through this approach. The main issue is that the existing works on this idea were focusing on demonstrating this possibility and not really fully characterizing the extent of leakage. So there were limitations in their statistical models that allow them to only analyze a smaller subset of eQTLs that are statistically independent. So this is the problem that we try to address, and we introduce this model that's working at the sequence level. So we're able to look at all the QTLs, accounting for their correlations as well. For those of you who are familiar with this, we're starting with this Lee and Stevens hidden Markov model, which basically puts a distribution over an entire genetic sequence, accounting for linkage disequilibrium. And we're adding another layer on top of this model, or on the bottom here, that's corresponding to gene expression variables, and we create these links that capture statistical correlation between genotypes and gene expression. And note that this is, again, including all the QTLs that are known. And we train this model in an end-to-end fashion so that the probabilities are calibrated to account for correlation between nearby genotypes. And the idea is that using this model, we can produce a match score for a given pair of gene expression and genotype profile. And this could be used to try to link individuals across data sets, which can potentially lead to re-identification. So in terms of evaluating this model, we took a data set. We had pairs of these samples, gene expression, genotype samples, from a set of individuals, around 300 individuals here. In this case, we included additional genotype profiles from a larger data set. In this case, it was haplotype reference consortium data. And this was a much larger set, so 22,000 individuals in the additional cohort. And we're trying to figure out, for a given gene expression profile, which genotype profile it corresponds to. And what I'm showing you here is the fraction of these individuals who were able to correctly link between these two samples as we add more of the additional candidate genotype profiles to the data set. And I'm comparing our method DSM to two other baseline methods, EBL and GMB. These are the previous approaches that I briefly mentioned that uses an independent set of eQTLs. The main thing to note here is that even after including this large number of individuals, so 22,000 individuals, we're still able to link around 90% or more of the original individuals based on gene expression information only. And our method is able to link a greater fraction compared to the baseline, and this gap becomes more pronounced if we consider a setting that's using a smaller set of eQTLs as well. So just based on chromosome 20, the overall numbers go down a lot, but the gap between our method and the previous approaches becomes clearer. The other thing that we can do here is that we can measure the confidence of our prediction of the links. And this is a metric that we developed. It's basically a p-value in some sense that measures the gap between the best match and the second best match. And if we compare this between our method and the previous methods, you can also see here that our method is on the y-axis, the previous ones are on the x-axis. The confidence that we have over the links that we produce tend to be greater. And what this leads to is that if we're working in a setting where we don't know if the matching individual is in the candidate pool or not, we have to draw a threshold somewhere, right? And have some number of false positives. And because of this higher confidence, we also get predictions that are that minimizes the number of mistakes compared to the true matches that we're able to find. There's a trade-off between precision and recall. And now I want to describe another recent work, which is illustrating a different type of privacy risk. And I'm bringing this up because I think this is a threat vector that not a lot of us have been aware of. So it's kind of a neat idea, I think, that we should all be aware of. And so here we're looking at the security of genotype imputation servers. I think many of you know about this. And so this is referring to services like TopMed and Michigan imputation servers. What they generally do is we provide a partially observed genotype profile from microarray genotyping platforms, for example, and then we obtain an imputed genotypes that fills in all these missing positions. And the way we're able to do this is by using a large reference panel of high quality genomes and using linkage disequilibrium pattern between nearby SNPs. And the question that we asked was, this process is using some information from the reference panel to be able to fill in the missing data, but how much information is actually being revealed throughout this process and included in the output? And in fact, can we actually try to reconstruct some of these sequences in the reference panel by interacting with these servers in certain ways. And just to avoid any doubt, we took great care to do this work responsibly and ethically. So none of this work was performed on these servers. It was all done in a local simulation environment. We also informed the stakeholders well in advance, actually almost a year in advance. And we're aware that additional security measures have been introduced after we published our work. And all the information that I'm sharing today is public and it's published last year. Okay, so the key idea that led to this work is actually the fact that if you plug in an input sequence that uniquely matches with a single reference sequence, it will extract the matching person's genome almost exactly. So this figure is showing that idea. So in this bottom left corner, you're seeing an example output from the imputation tool. We're showing it as predicted dosages, which is a value between 0 and 1. And you can see this value across the genomic window that's being imputed on the x-axis. If there's a single match, the predictions are very confident. It's near 0, near 1. And this corresponds to the actual genotypes of the matching person. What's even more surprising is that if you have a match, have a query that matches a small number of reference samples, you can actually see this unique pattern that corresponds to that too. So if it matches 3 samples, then you'll see these predicted dosages that are aggregated around multiples of 1/3. So what this means is that you can look at the imputation output and figure out how many matches there were in the reference panel. And what this leads to is this type of potential attack pipeline where we start with a lot of random queries, impute them, and see what we get in terms of these dosage outputs, and figure out which one of them was unique, which ones had multiple matches. And for those with multiple matches, we can try to extend the query to make it more unique. And in the end, we pull together all of the unique matches, and this corresponds to a set of haplotype sequences from the reference panel. And using 1,000 genomes data as the reference panel, as an example, we were able to demonstrate that this type of attack can actually extract a fair amount of sequences from the reference panel. So for example, if we impute 1 million input queries that we constructed in some way, we're able to extract almost 80% of the sequences in the reference panel. There's some amount of error that's happening here because of the misclassifications in the pipeline, but this error rate tends to be fairly low. And here's a slightly different setting where, to try to prevent this attack, what we could do is instead of releasing the predicted probabilities, we can release just the discrete genotype predictions. So either 0 or 1, nothing in between. But even in this setting, there is a workaround that allows us to extract unique matches. So the general idea is that you can take the output sequence, introduce additional changes, or keep a subset of positions, mask again some of the other positions, impute it again. If the result stays the same, then it's more likely that the original sequence that was output was corresponding to a unique individual. So using this type of idea, we can still extract sequences at slightly lower effectiveness, as you can see. One big caveat here is that the sequences that we're getting here are chunks from the private genomes. It's not the entire genome. And this is because imputation pipeline is run independently on smaller genomic windows, typically around 20 megabase pairs. But it turns out that you can start from these fragments of the genome and link them across different windows using relatedness patterns. And this is just a visualization. I won't go into details here, that if you compute the kinship coefficient between these individual pieces with relative of a certain degree, you can see that the kinship is separated from, is far enough from 0 that we can distinguish it from unrelated samples. And what this means is that we can imagine a setting where we have access to a secondary data set, which might potentially include relatives of the individuals who are in the reference panel. And then we could compute these kinship coefficients. It's written as semi-kinship because it's a modified version that's working with haplotypes instead of diplotypes. So we can compute these kinship coefficients between pairs of samples between these two sources. And then we feed this into our probabilistic linking algorithm that's taking this matrix as input and then outputs groups of sequences that are likely to have come from the same individual. So that's the overall outline of the attack vector. And we were able to show that given a data set with real relatives that are known to be included in the relative set, we were able to link a substantial portion of the target genome up to third-degree relative. So beyond that, the signal becomes too weak. So we can't link too many samples. And based on this data, we can extrapolate to get a sense of what fraction of the genome could be linked for different proportions of the reference panel individuals, given different sizes of this relative set that I was referring to, relative to the overall size of the population that the reference panel came from. So for example, if we're given a relative set that includes 0.5% of the underlying population, we could expect to link 17% of the genome for at least 5% of the individuals in the reference panel. So that's one of the estimates that we were able to get. And this is a realistic number given that 0.5% is comparable to the size of the UK Biobank, for example. So just to summarize this portion, I've shown you different types of genomic privacy risks. We looked at re-identification attacks, membership, phenotype inference, data linkage, as well as data reconstruction. And the overall takeaway is that our understanding is still rapidly shifting. And this needs to continue to change as we gain access to different types of data, different models, more advanced AI and ML tools as well as other types of data sharing systems that are emerging. And given all these changes, it's important to have these rigorous approaches to analyze these risks and assess them using new models and come up with new ways to address these risks. So on that point, in the second part, I'll give you some ideas of some of the emerging technical solutions for addressing these risks. I think you're all familiar with these existing policy frameworks like common rule privacy, HIPAA privacy rule, GDPR. They define these notions that we all know and love, like IRB review processes, informed consent, the idea of protected health information. And there's no question that these frameworks provide an important tool for us to safeguard the use of biomedical data, but it's also important to note some of the inherent limitations to these regulatory approaches. So the first thing is that designing and updating these regulations is generally a slow and long-term process for a good reason. One example is that genetic data was classified as protected health information for HIPAA only in 2013, several years after genetic data have become more widely available. There are also these key ambiguities in some of the terms that we use in these regulations, right? So when we say de-identified or anonymized data, what does that actually mean, and how does it apply to the data sets that we're actually dealing with? And as I've demonstrated, hopefully, a lot of the biomedical data types, it's not possible to completely de-identify it. There's always going to be some amount of identifying information, sorry. So we need to draw a line somewhere when we think about laws and regulations. And perhaps the most important pitfall is that these approaches don't resolve the core conflict between the need to share data and the need to protect privacy. And this is because they're typically about limiting the scope of data sharing and usage, depending on the context, in order to provide privacy. So it turns out that there's a collection of techniques from the computer science literature, which are referred to as privacy-enhancing technologies, that can help us resolve this conflict. And at a high level, these provide us with mathematical techniques that can help us use and share private biomedical data while at the same time protecting privacy. So this is what I'll tell you about briefly next. I'm going to highlight 5 of these technologies that belong to this category. I'm going to start with homomorphic encryption, which is a type of encryption technique. And it's a special form where the structure of the encryption allows us to perform operations directly on the encrypted data without having to decrypt it first. Initially, these frameworks were limited to performing limited types or limited numbers of operations on the private data. But there's been remarkable progress over the years, and now we have these practical systems that can help us implement fairly complex analytic tasks. But they do come with a computational overhead, because now we're operating with encrypted data, And that's a lot more expensive than working with non-encrypted data. A slightly different approach is secure multi-party computation. It has two acronyms, MPC or SMC, depending on who you ask. And the core of this framework is not encryption, but we're going to use this technique called secret sharing. And what it means is for each private number, we divide it into a set of random numbers that somehow all add up to the private number. And we're going to split them up and then distribute it to multiple parties. So they all together know, well, the shares collectively encode information about the private number, but if you look at each party's information, they don't know what the private number is. And the idea is that we can design these interactive protocols where these parties work together to perform computation on the underlying secret without revealing anything throughout this process. So at the end of a protocol like this, we get the output of the analysis results as secret shares as well, which we can combine to reveal the output. So this is not using encryption. So the computational operations tend to be cheaper than homomorphic encryption, but at the cost of greater communication burden. So that's the trade-off. Trusted execution environment, or TE, it's a hardware approach to secure computation. The most well-known examples are Intel SGX and AMD SAV technologies. And what they provide is a way to create these isolated environments in a computer that protects the data inside it confidential, keeps it confidential, even from the person who's operating on these machines. And the idea is that we can use this to send private data into this environment, isolated environment called secure enclave, do some processing on them, and return the results to the user through encrypted channels so that no private information is leaked during the process. The key feature of these technologies is what's called remote attestation. And this is the use of cryptographic signatures that actually allows us to verify that the enclave environment has been created faithfully, and also the fact that the program that's running inside it is what we initially assigned to it to run. So in this environment, we're working with non-encrypted data. So there's no computational overhead here, or very minimal computational overhead. But this comes at the cost of relaxed privacy notion, since we're relying on a secure hardware component. You've probably heard of federated learning. This is popularized by Google several years ago, initially designed for this use case of training machine learning models across millions of mobile devices. The idea is fairly simple in that it's, we have a model, and then we compute these local model updates on each device, aggregate them across devices, and apply the global update to the shared model and redistribute them. And we're repeating this process to improve the accuracy of the overall model. This has immediate extension to the biomedical setting where we have a smaller group of collaborators who can also try to jointly train machine learning models this way. The focus so far has been mostly on machine learning models or deep learning models that can be trained with gradient-based optimization. So the relevance for some of the genomic analysis tasks may be limited because we're using different types of statistical models, but it has found really useful application in medical image analysis domain where we do need these deep learning models that are trained with gradients. And this aggregation of local updates, this can reveal some private information from each party that's participating in this workflow. So this needs to be carefully considered. And typically, these methods are combined with other techniques like encryption or differential privacy, which is what I'll mention in the next slide, to increase the security level of this solution. So differential privacy, it's addressing a slightly different problem where we're concerned with the leakage of private information in the data that is released at the end of the computation. So we can think about GWAS statistics, for example. The key concept here is this notion of neighboring databases. So these are two hypothetical databases that differ in exactly single individual. And the idea is that we want to introduce some noise into the system or into the computation so that only based on the released data, we can't really distinguish between these two neighboring databases for any two neighboring databases. And this privacy guarantee is typically controlled by this privacy parameter epsilon. And in this case, values of epsilon that are closer to 0 means higher privacy, and greater values mean lower privacy, but at the same time, it allows us to introduce a smaller amount of noise. So the results tend to be more accurate. So this framework has been applied to GWAS statistics release and other types of biomedical database queries, like looking for cohorts that match a certain criteria. But for many of these existing studies, they were limited to specific, simpler analysis settings. For example, for the case of GWAS, instead of releasing the entire genome-wide vector, we can only release, say, like the top K most significant hits or something like that. That's been the typical setting that's been studied. And the main reason is that the actual noise that we need to introduce to achieve differential privacy guarantee tends to be very high in high dimensional cases. So these techniques are useful in a range of different settings in biomedical research. I've listed out some examples here that we've worked on over the years. Just to mention at a high level, we can use these tools to build softwares that can help researchers across institutions run collaborative studies in a secure way. We can also build analytic services that take users' private data, process them, and returns statistical insights back to them, all in a privacy-preserving way. There's also this private data release setting, similar to what I just mentioned with GWAS, where we want to release some privatized version of a biomedical data or analysis results with some meaningful guarantees of privacy for the underlying individuals in the data set. And we're not the only ones working on this. There's a growing community of researchers developing these tools. I just wanted to mention a couple of examples of these real-world competitions where we bring people together from different communities to create solutions for these tasks. So iDash is a popular one in this domain. Last year, there was also a governmental collaboration between US and UK on developing tools based on these technologies. One of the tasks was creating a model for pandemic forecasting. And some of us at Broad entered these competitions and had winning solutions for both of them. So now I'll briefly go through some of our recent work in this domain as well. So in our recent work, we introduced this tool called Secure Federated GWAS, or SFGWAS. This is a cryptographic approach to performing GWAS across multiple institutions without sharing any private information between them. The key technical insight that we introduced in this work is that, as opposed to using a single technology, one of the ones that I mentioned earlier, we can actually use a combination of them to gain computational speedups, which tend to be quite important in practice. So in this case, we're combining MPC and homomorphic encryption, which allowed us to design these federated secure computation systems. And what that means is that instead of encrypting the entire input data set and sharing it between the parties, we can keep all the local input data sets local and only share encrypted versions of intermediate analysis results to carry out the global computation. This reduces communication and also speeds up computation at the site level, because now we're able to use unencrypted data. And we were able to show that this results in an order of magnitude improvement over the prior art that was only using MPC technology across these different GWAS databases. And this scalability improvement allowed us to take these tools and then apply it to really large biobank scale data sets. Here I'm showing you two examples, eMERGE and UK Biobank. So the latter in our experiment included around 276,000 individuals. We're splitting these resources across 6 or 7 simulated federated units to demonstrate this ability to perform joint analysis without sharing private data. And I'm showing you the results of GWAS that matches accurately with the centralized analysis, where all the data is pulled together into a single location. And the overall runtime for these pipelines tend to be around a few days for databases of this scale. So this is not a cheap pipeline to run, but the runtimes are still practically feasible, and these can be brought down further by introducing additional computing resources. And I want to point out that the experiment that we ran on UK Biobank is actually 2,000 times larger than the previous benchmark that we tested in a previous study. And also want to highlight that in this initial pipeline that we demonstrated, we're looking at PCA-based workflow, where we run principal component analysis to look for these ancestry covariates to include as fixed effect term in the GWAS model. But it's also common to replace this with a random effect model, which is known as linear mixed model approach to GWAS. And this is generally regarded as being more accurate for GWAS. I don't need to tell you this, but it also comes at a greater computational cost, even without any encryption. So we weren't able to do this using secure computation techniques for a long time. But recently, there was this method that was introduced called Regini. And it's doing an efficient approximation of this random effect term using a whole genome regression model. And what this allowed us to do is distribute this computational workflow across the parties using our cryptographic tools, and also obtain a practical pipeline for running LLM-based GWAS. And the distributed technique that I alluded to turned out to be fairly significant. So we were able to remove dependence on the size of the cohort that goes into these GWAS studies, as you can see in these runtime plots that stay nearly constant across different data set sizes. We're able to show that the output of our LLM analysis workflow closely matches with a centralized execution of this Regini tool on the pooled data set. So now we're able to run these different types of GWAS workflows in a secure and federated manner. We had this related work more recently, which was about finding genetic relatives across different data sets that cannot be pulled together due to privacy concerns. And this is a key pre-processing step in many studies or federated databases like Nomad, where the presence of these relatives could skew the distribution of the statistics that we want to compute and release to the public. So we have the cryptographic tools to be able to compute this relatedness coefficient privately, but the main challenge here is the fact that we need to perform all pairwise comparisons between these two data sources, and that turns out to be very expensive. So the key idea that we had in this work is that we could hash and bucket the individuals so that we can perform comparisons only between individuals that belong to the same bucket. And the idea is that due to identity bias and sharing between close relatives, the close relatives have a higher probability of getting assigned to the same bucket. So this still retains high accuracy. We're able to demonstrate that on UK Biobank and all of us data. You can see that this overall recall for detecting up to third-degree relatives is very high. It's 97% or higher in all three cases. The precision is also higher than 98%. And some of these false positive findings are arguably not false positives. What they are is that they have a relatedness coefficient that's really close to the threshold, and due to numerical precision, it's crossing that boundary. And the overall runtime was also practical, less than a day for all these data sets. So where are we headed next? I'll try to conclude the talk in the next few minutes. So I think these technologies are maturing enough that now we're able to see these inklings of practical tools for different genomic analysis tasks. And I think over the next few years, we'll see a lot more of these emerging for different analysis workflows, including things like find mapping or predicting disease risks based on personal genome. So we're actively working on some of these ideas. And I think these would be valuable tools for people doing genomic analysis across different sites to help with collaboration. And we are currently working on a deployment study where we're trying to run some of these joint analyses between Million Veteran Program and All of Us research program. So as you know, these are two of the largest biobanks in the US. They're also known for their strict security guidelines that prevent the data from being shared externally. So we're trying to use our tools to create a link between them to enable joint studies. Last year, we launched our SFKit web server. This is joint work with Broad Data Sciences Platform. And this is a web-based service that allows people to run secure federated tools, the cryptography-based ones that I mentioned, in a push-button kind of way. And they can bring their own data set, run a collaborative analysis with their collaborators using the service, all the components that we have here are open source. And this is actually the service that we're using for the ongoing pilot study. And currently, we're also working on integrating this set of tools into existing cloud-based analysis platforms like Terra, which is also developed at Broad, as well as all of us researcher workbench, which is based on Terra. So these are some of the things that are coming up down the road. And lastly, I'll just mention that there's an upcoming conference that would be of interest to all of you. So Recom is happening. It's being organized at MIT. How convenient. And there's actually this inaugural satellite conference on biomedical data privacy and equity, which is happening one day before the main conference. And we have several talks in both of these venues. If you're interested, please check out the programs. And you can also reach out to me if you want to discuss more. And with that, I want to thank all of the former and current lab members of my group and my collaborators and mentors in different institutions for all their input and support. Without them, we couldn't have done a lot of these works. And I thank you for your attention. I'll take any questions.
Great. Thank you so much, Dr. Cho. I think we have time for one or two questions. I might start them off. So if we think about how, like each of us, we're all required at publication to deposit all of our transcriptomics, right, in a public data frame. And I try to think of how we implement this. Does it mean that like the gene expression omnibus data, like should it be at the repository level that these things are implemented so researchers could still upload their kind of naive data frames as we do currently, and then the implementation of privacy happens at the repository? Or do you think this is actually something that where even that step is not secure enough and it should be, the privacy should be implemented prior to upload?
So I can imagine a repository like Geo being run like something like dbGaP with the genotype data with stricter access control mechanisms. I'm not necessarily proposing that needs to happen, but we do have some concerns about the way currently transcriptomic data is being shared because they do leak genotypic information. And it's not just our concern. There are other people who's been pushing on this as well. And I know that there's an ongoing conversation with NIH on this topic. So yeah, there might be some changes in the future, but we're not sure yet. I do want to make a note that the risks that I showed about that data is kind of in a controlled environment, right? Where we have this matching ancestry between like the target individuals and like the other individuals in the candidate pool. So there are like other considerations like that that needs to go in when we actually think about the risk in the practical setting.
Makes sense. Great. Well, thank you so much for a broad sweeping talk that really highlighted, I think, so many different ways in which privacy impacts the research that we all do every day. And then please feel free to grab second breakfast, and then we'll start the next session at 9:30.